The good thing is it's French, so it doesn't interfere with the English. Great, that is set up and running. Well, welcome everyone to Neuroccino on this Monday morning, the 14th of June, 9:30. We have one paper today presented by Valentina to us, and I happily give you the floor in a second. Just brief recap. The rules is everyone is welcome. Just bring a paper that got you excited. Tell us why it got you excited, and we can talk about it to the best of our knowledge, and then we discuss it as well. With that, Valentina, whenever you're ready, you can share your screen and take it away.
Voila. Okay. Hi, everyone. So today I'm going to present a preprint by Sophie Volk, who's been a guest to the CNS seminar in the previous session of the CNS seminar. I really recommend you to go and listen to her talk again. So this paper is on the genetic and phylogenetic uncoupling of structure and function in human transmodal cortex. Oops. I don't know what's happening. Sorry for the screen sharing. Okay. Can you see? Looking good. Looking good. Looking good. Okay. First of all, difference between transmodal and unimodal systems in the brain. So transmodal systems are referred to highly cognitive functions and functions that are engaged in abstract processing and self-generated cognition. While unimodal systems are more related to sensory, for instance, sensory and motor regions that are specifically dedicated to one modal cognitive function. There are differences also in the brain structures who support transmodal and unimodal systems. In particular, unimodal sensory and motor regions rely on distinctive lamination patterns, while transmodal cortex appear to have a more agranular and dysgranular pattern and less apparent lamination. Furthermore, transmodal regions are lower in myelin and have a more complex dendritic urbanization, probably to facilitate the integrative processing, which is fundamental in transmodal systems communication. The differential organization between transmodal and unimodal systems reflect also on the coupling of cortical structure and functions. In particular, it's been observed reduction in the constraining of the mapping of transmodal functions on brain structures. And this is probably important for the evolutionary adaptation supporting specifically human cognition. But the author's goal was to map the structure-function relationship across the human cortex and to explore whether these profiles are hereditable. Furthermore, and this hypothesis is supported by previous studies who showed that microstructure and functional connectivity are indeed partially heritable. And furthermore, they examined how structure and function association is different between human and non-human primates. So let's go to the study. The first step, as I was saying before, of the study is to observe the coupling between the function and the structure of transmodal and unimodal systems. To do so, the authors computed the maps of functional connectivity and a cortical microstructure. In particular, they computed these MPC microstructure profile covariance matrices, which has been sampled via T1 and T2 values at 12 different cortical depths. And then for the functional connectivity part, the authors calculated resting state functional connectivity matrices that I will refer to these two as structure matrices and resting state matrices along the study. And so they computed these resting state matrices by cross-correlated the neural time series between pairs of 400 cortical nodes. But I want to show you the figure. Maybe it's easier. So these are the microstructural profile covariance, and this is the computed resting state matrices. Then they observed the coupling between the structure matrices and resting state matrices via correlation. And they have seen that we have the highest correlation for unimodal systems, while we have weaker correlation for transmodal systems. Here are the cytoarchitectural class of the system. It goes from idiotypic to unimodal, heteromodal, and paralimbic. And then the author explored genetic control over the structural and functional connectivity profiles. They are using the Human Connectome project dataset to provide also information on the genetic similarity, familiarity in the dataset. So they computed node-wise heritability of the structural matrices and correlated the heritability with the mean structural matrices to have an index of genetic control. And they did the same thing for the resting state matrices. And in this case, higher correlation suggests that the structural and resting state functional correspondence show also high heritability and in the opposite for lower correlation, so lower heritability. And also here I want to show you the figures. The figures show that there is higher correlations for and this means higher heritability for primary regions, and weak correlation for unimodal association cortices, and zero or closer to zero correlation for higher transmodal cortices. So in a further step, the author looked at the correspondence between the structure and function in non-human primates, and they used the primate data exchange repository And they computed the structural matrices similarly to the approach used for humans. And then they carried out the functional analysis and aligned the functional analysis to for human and macaques. And then they compared the structural and functional matrices for the macaques and found a correlation between here they found a correlation between the structural and functional matrices similar to those of the humans. They see a weaker correlation for transmodal cortices and higher correlation for unimodal ones. Okay, and so both unimodal and heteromodal regions were found to be more coupled in macaques compared to the human results that I've shown you before. Then they run an analysis to observe the differences in microstructure and functional connectivity. And now I need you to be really patient with me because I had an hard time understanding this kind of analysis, who involves the gradient observed in structural and functional matrices. So they evaluated the correspondence in the organizational axis of structural and functional heritability in a node-wise, so the 400 nodes. They applied the nonlinear dimensionality reduction and observed that structural and functional matrices followed similar spatial axis compared to their heritability. Then they calculated the nodal differences between the standardized and the aligned structural and functional gradients, and they observed that the gradients were most uncoupled in the transmodal systems. Then they selected the homolog gradients of structural and functional matrices in macaques. And again, they observed that there is the highest uncoupling within the transmodal systems. Finally, the authors looked for the functional outcome that the structure function decoupling will cause. And so via always dimensionality reduction, they plotted the gradient differences in both humans and macaques. So on the y-axis we have the coupling, and on the x-axis we have the difference between the resting state and structural gradient. And comparing human and macaques, we see that the upper quadrants here, they both include sensory and motor unimodal networks. But in macaques, the left upper quadrant, so this one, it contains also frontoparietal and the thought mode networks that are in the lower left quadrant in humans. The lower right quadrant is contains the limbic and ventral attentional in humans, and only the limbic network in macaques. And if they compared the relative numbers of nodes distributed in each quadrant, and they observed that for the humans, more areas were uncoupled when compared to macaques. Furthermore, according to looking at the dual origin of the cortical differentiation model. So the authors suggest that the differentiation of the cortical reorganization reflect the support the dual origin organization of the cortex. And they looked also at the prenatal and prenatal and postnatal organization, and they see that there is a trend that goes from the hippocampus and thalamus and amygdala for the humans, and the hippocampus for the macaques. And finally, they projected via meta-analytics map retrieved from Neurocino. They projected the meta-analytic maps on the reducted space to see how they rearrange themselves, and they see that the lowest uncoupling, so the lowest coupling functions are related to biographical memory, social cognition, and episodic memory. And according to the author, this suggests that this makes us unique compared to the macaque, and because in humans the uncoupling of these functions means that they are more unique to us in evolution matter. So it was a really difficult paper for me to understand, but I was amazed by this analysis that space from genetics to species differences in humans and macaques.
Lovely. Thank you, Valentina. Very, very great presentation. Thank you. Dense paper, but very exciting indeed. Thank you. I can see that Michelle has a question.
Do you wanna unmute yourself, Valentina, because we're next to each other? Sorry. So I got three questions actually. One which is a bit general, and that's a very impressive gamut of different methods around the concept of brain organization. But I don't see how altogether they help us to have an overall, if you had to summarize the paper in one sentence, what is the big thing that the paper brings up?
To me is that is really, I mean, to me, the most interesting and striking result was this one. So that the uncoupling of autobiographical and like social cognition functions it is what makes us unique as humans. And you see that evolutionarily the brain structures reorganized according to our ability of thinking about our past and ourselves, and socially interact with the others.
Okay, which brings me to my second question. I mean, we can say that elephants have a very good episodic memory, so making it unique to humans. I don't know. And I just like so this interpretation of this is unique to human is really based on the comparison of embedded space that have been built separately. And if they've been built separately, I was wondering how stable those spaces are, because we've been playing with that, and we know that like when you rerun your embedding space, or you run it with different data, the shape and the organization can change even if you're measuring twice the same thing.
No, I'm sorry, I'm stopping you because maybe I wasn't clear. I skipped that part. The embedded space was always the same. They plotted the maps on this original embedded space.
And then last question, really, which is based on the genetic relationship of the functional and structural organization. I couldn't help but see that, and that's a previous figure, I think, that areas that actually are equal to a little bit before, I think, yes. So areas that have a R of 0, which would be there is absolutely no relationship between, it's not even an inverse relationship, there is no relationship at all between twins. Those areas are actually the areas that we report as being the most variable in the brain in people that are unrelated. And so I was wondering whether, is this really related to gene and similarity, or is there a latent variable here that would explain the results, which be related to how nature makes the brain variable?
So if I got your question, is like you say, it's not just a matter of genes, it's also a matter of evolution. It's but they are related to each other. I mean, something that it's hereditary, it's also like selected by evolution. It's hereditary because it's evolution.
What does mean is like we have the same map but with unrelated people. But just entire individual variability. Sorry, it wasn't clear.
No, but it makes sense. It makes sense because the less hereditary. So these are the structure, like the higher cognitive transmodal system structures. So I mean, it makes sense if you have maps of non-related individuals compared to these ones, because this means that we have a stable and there is less interpersonal differences in unimodal regions respect to the transmodal systems. And this is what the paper shows here. Like it's and you have the cause of this interindividual variability. It's genetic.
Can I jump in right there, because we have a question in the chat that actually is relevant to where you're going at the moment. And the question is, when you say genetically related participants in the HCP, are we referring to twins versus non-twins?
Sorry, I muted myself. And it's twins, if I remember well. It's they observed the twins pattern.
Okay, thanks. Because when you say like I was reading briefly the text and I just read genetically related, so maybe I thought maybe I didn't know, but if the ACP they have some genetic sequencing, but probably it's just twins.
I'm not sure. I have to check the methods again, but.
Yeah, because I where you were presenting, I was reading and there wasn't the word twins, so I was wondering. But yeah, thank you. There was the release, so we'll check.
Yeah, I think it's twins.
Proper multitasking, Leah, listening to Valentina and reading the paper at the same time. I'm impressed. You unmute, but that's fine. Are there any other questions in the room? No. Okay. So it's a lot to unpack, I guess, in that paper. Different species, different methods. It's quite impressive. What are the limitations that you can see or that the authors discussed in the study?
So I didn't get to the limitation actually. It's a really long paper, and it was very dense to me because these are not subjects that I'm used to, and I'm learning like right now, so it's part of my postdoc training. So the limitation of the study, I guess like using all these combined methodology, all these diverse methodology, and apply them in the human macaque differences. Actually, I had enough hard time to understand those, and I don't know. I'm not 100% sure that we have a clear match, you know, between of comparison between the two species, especially when using dimensionality reduction. With, I mean, it has its benefits, but also you reduce the information that you have, and yes, we are talking about two different species, human and non-human primates. I don't know. I have to go through this maybe another time, a couple of times more.
Fair enough. Another question I had, if you could go back to the graph you showed that was of greatest interest to you with the uncoupling and the cognitive dimensions.
I don't know why you're presenting the, okay, it's 2 pages.
I can show you later when I come to the office.
Thank you. Oh, this is the first time it happened to me. Okay.
You're using Preview, right?
Oh, what?
Are you using Preview?
No.
Okay. Well, we look at that later and fix it for the next presentation. Yes, so that figure, so you talked about memory in the bottom left corner for the uncoupling, but I was wondering if the quadrant actually is meaningful, because you would also have language in there, and is that just a quadrant that would make us unique, or particular memory because that's further?
No. Sorry, I mean also language is in the left bottom corner. So yeah, no. Also the language functions. And
so that's the bias between you and me. You're focusing on memory and I'm focusing on language.
No, thank you for the remark. It's really important.
It just stuck out to me, but I guess that's just me being interested.
We complete each other.
We do. Okay, thanks for clarifying that. Any other comments that came up in yes, there's a question in the chat. Are there any evidences of postmortem or histological information of these different kind of cortex, unimodal and transmodal cortices that agree with the results that you presented from the paper?
Yeah, the authors discuss like straight away in the introduction of postmortem. Yeah, here there are postmortem evidences. Sorry. Yeah, in non-human animals and both non-human and human species, yeah. So post-mortem evidences show that similar regions with similar seat architecture are more likely to be structural and functional interconnected. And then we can also go through the discussion to see if they talk about post-mortem studies. I don't remember. I'm not sure. I'm not sure, but I will go through the discussion again and maybe text you later. Who wrote the
I wrote the question, sorry, I'm writing today. No, it was like I was asking, trying to find a link between all the advanced analysis that we have in imaging and another modality comparison. But I can see the discussion is really wide and maybe
yes.
somewhere.
Thank you. If there's no further questions, I suggest we all read the paper to answer our questions. And also, if there's anything that we can't ask or something more specific, Sophie is very approachable. So just volunteer her time here, but just get in touch with her. I'm sure she's happy to talk about the paper with you directly as well. Thank you so much for presenting the paper, Valentina. Absolutely exciting study indeed. And as this is the only paper of today, we are at the end of Neuroccino for this Monday morning. Whoever wants to present next week, feel free to just bring a paper you get excited about and talk to us about it, primarily why you got excited about it and what the authors did, and then we discuss it as you can see today. With that, I wish you an exciting week in science, and we see you next Monday. Have a good week. Bye.